Hi everyone. Today, we're going to examine some new research that details a specific molecular mechanism of aging. It looks at how centromeres, which are the structures critical for chromosome segregation, become inactivated over time. And it also explores a potential strategy to reverse this process.
So we'll start with the core observation of centromere decline in aging. From there, we'll get into the molecular pathways involved, looking at the p53 connection, and then uncover an epigenetic repression mechanism. After that, we'll explore a dual inhibition strategy for reactivating the aged centromere, define the role of CENP-B in this whole process, and wrap up with how these findings were validated in human tissues.
The first step in this investigation was really to establish whether centromere integrity is actually compromised during human aging. To figure this out, researchers performed a broad analysis, looking at both gene expression and protein levels in aged human cells and tissues.
The researchers started by analyzing the Genotype Tissue Expression, or GTEx, database. This database contains transcriptomic data from thousands of human tissue samples. When they compared tissues from older individuals, in the 60 to 79-year age group, with younger ones, they found a very distinct pattern. There was a widespread downregulation of genes related to the kinetochore, which is the essential protein machinery for chromosome segregation. Now, critically, this included CENP-A and CENP-C, two foundational proteins for centromere identity and function.
Okay, so that's at the RNA level. The next question was whether these changes translate to the protein level. To find out, the researchers performed immunofluorescence on skin fibroblasts from donors of different ages, and the results did confirm the trend. They saw a significant drop in the centromeric protein CENP-C, a key binding partner for CENP-A, starting in cells from a 44-year-old donor. This confirmed that the actual protein components of the centromere are diminishing with age.
Quantitative immunoblotting give a more precise measurement of this decline. In whole cell extracts from a 75-year-old donor, the protein levels of both CENP-A and CENP-C were less than half of what was found in a 1-year-old donor. A reduction of this size strongly suggests that the structural integrity of the centromere is substantially weakened during the aging process.
So now that the phenomenon of centromere decline is established, the next question is, what's the underlying cause? The investigation turned to a prime suspect in cellular aging, the tumor suppressor p53, which is a well-known regulator of the cell cycle and senescence.
In laboratory models of induced cellular senescence, a clear inverse correlation was seen. As p53 protein levels went up, levels of the key centromeric protein CENP-A went down. To test if this was cause and effect, the researchers used siRNA to silence p53 in aged cells. The intervention was successful. The protein levels of both CENP-A and CENP-C were restored.
This, however, created a bit of a paradox. Even though the centromeric protein levels were returned to normal, the cells continued to fail at cell division. They still showed a high frequency of mitotic errors, like lagging chromosomes and cytoplasmic bridges. This persistence of mitotic defects, despite the recovery of CENP-A and CENP-C, suggested that another regulatory mechanism was at play, one that was independent of protein abundance. The fact that these mitotic errors remained suggested the problem wasn't just a lack of building materials, but maybe a flaw in their assembly instructions. This led investigators to look at the epigenetic state of the centromeric DNA itself. The investigation turned to non-coding centromeric transcripts known as hsat-tRNA. These transcripts are essential for recruiting and loading new CENP-A onto centromeric chromatin. The analysis showed that hsat-tRNA levels were significantly repressed in aged cells, and critically, silencing p53 did not rescue their expression, which confirmed that the regulatory block was happening at the level of transcription. To find the source of this transcriptional silencing, they performed chromatin immunoprecipitation. The results showed a definitive shift in the epigenetic landscape at centromeric DNA in aged cells. There was an increased occupancy of H3K9ME3, a canonical mark of repressive heterochromatin, and at the same time a loss of H3K4ME2, which is a mark associated with active transcription. This demonstrated that centromeres are in fact epigenetically silenced in aged cells.
The discovery of this specific epigenetic modification, the loss of the active H3K4ME2 mark, pointed to a particular enzyme as a potential target. The idea was that by targeting it, they might be able to reverse the transcriptional silencing.
The enzyme implicated here is lysine-specific demethylase 1, known as KDM1A, or LSD1. Its job is to remove that H3K4ME2 activating mark. So the researchers hypothesized that inhibiting KDM1A activity at the centromere could reverse the epigenetic silencing and, in turn, restore centromere function.
This led them to design a dual inhibition experiment. First, p53 was silenced in aged fibroblasts to address the deficit in CENP-A protein. At the same time, the cells were treated with 2-PCPA, which is a small molecule inhibitor that targets the catalytic activity of KDM1A. This two-pronged approach was designed to correct both the protein level and the transcriptional level defects.
And the outcome of this dual inhibition strategy was a functional rescue of mitotic fidelity. While silencing p53 alone wasn't enough to correct chromosome segregation, the combined inhibition of p53 and KDM1A led to a significant reduction in the rate of mitotic defects. It actually restored fidelity to levels that were approaching those seen in young fibroblasts.
This molecular rescue also had a direct functional consequence. Cell proliferation was measured using EDU incorporation, which marks cells that are in S phase. The dual inhibition treatment resulted in a twofold increase in the population of proliferating cells compared to untreated aged cells. This indicates a restoration of the cell's proliferative capacity.
So, one piece of the mechanism was still missing, identifying the factor that's responsible for recruiting the KDM1A repressor enzyme specifically to the centromere in aged cells, which is what would initiate the epigenetic silencing in the first place. The evidence pointed to centromere protein B, or CNPB. Now, unlike CENP-A and CENP-C, CNPB protein levels don't decline with age. When CNPB was knocked down in aged cells, the expression of centromeric HSAT RNA actually increased, which implicates CNPB in a repressive role. Furthermore, proximity ligation assays showed a significantly increased interaction between CNPB and KDM1A in aged cells. This supports a model where CNPB is what recruits the epigenetic silencing machinery. Finally, to make sure these findings weren't just an artifact of cell culture, the study sought to validate the observations in post-mortem human tissue. They also wanted to assess whether the mechanism is conserved across species. And the analysis of human kidney and lung tissues from donors of various ages did confirm the key molecular signatures. With advancing age, both tissues showed a clear decline in CNP-A protein and a reduction in centromeric HSATA transcripts. And notably, in kidney tissue, CNP-B expression was seen to increase with age, which is consistent with its proposed role as an age-dependent repressor. This phenomenon also appears to be conserved across mammals. An analysis of a mouse aging transcriptomic dataset revealed a parallel decline in the expression of essential kinetochore genes in older mice. This suggests that centromere weakening may be a fundamental and conserved aspect of the mammalian aging process.
So, to summarize, this study presents a cohesive two-part model for centromere inactivation during aging. It involves p53-dependent protein downregulation and CNEKB/KDM1A mediated epigenetic silencing. The successful rescue provides a proof of concept for its reversal, which raises an important question for future research. Could targeted epigenetic reactivation of centromeres represent a therapeutic avenue for mitigating age-related cellular decline?